Hello, and welcome to this tutorial developed by the University of Leicester Genetics Department. This program will model phase variation in bacteria and help you visualize the concept covered in the tutorial. On the screen, you can see four options. These represent four different mutation rates and will determine the probability of phase variable switching in the simulator. The bacteria in the simulator have three phase variable genes that can be switched on or off interchangeably by mutation. On the screen, you can see a schematic representation of each of the three genes. At the start of each gene is a poly-G tract. In this case, when 5 Gs are in the tract, gene expression is switched off. Insertion of another G into the tract will cause a frame shift mutation. This switches gene expression on, allowing functional protein molecules to be synthesized. As we know, phase variation is reversible, and a deletion in the poly-G tract will reverse the process. It is also important to remember that polynucleotide tracts are not always made up of Gs, and they are not always 6 bases. Phase variation of each gene can occur at different times and in different orders. One gene may switch on, while the others do not. Two may switch on, whilst one does not. And in rare cases, all 3 genes will switch on at the same time. Okay, so now let's take a look back at the simulator. The simulator will begin with a single bacterial cell. This cell will divide into 2, and those into 4, and so on for several generations. Each time a cell divides, there is a chance that a phase variable switch will occur. These switches will occur in different orders and at different times, given a mix of different genotypes. Each gene is represented by a number. The first number in the set of 3 represents the first gene, the second number, the second gene, the third number, the third gene. If the gene is switched on, the number will read 1. If the gene is switched off, the number will read 0. For example, cells with only the last gene switched on, or the 001 genotype, will appear dark blue, whereas cells with all 3 genes switched on, the 111 genotype, will appear as white. Run-in totals of each genotype in the population are given beside the respective colour in the key. As the simulator progresses, you can see the genes switching on and off through the changes in the colours of the cell. Once the simulator has finished, you can scroll back through the sequence of events using the bar at the bottom of the screen. This will help you see individual changes in specific cells at any one time. Now we've learnt how to use the simulator, have a go at the questions below. Thank you, and we hope you enjoy.